Okay.
They do it themselves.
They do it themselves. Thank you very much, Kara. You're coming from the parent perspective, and you mentioned these 6 groups, the advisories, the stakeholders, industry, regulators, payers, caregivers, and patients. And we're now sort of flipping sides. Dr. Patroula Smpokou comes from the regulatory side of the coin. And she's working at the FDA's Center for Drug Evaluation and Research. So she's going to explain to you what their part of the whole business is. And every patient, and sometimes even the scientists, including me, do have the impression those are the guys that just cost you time and money, and especially time is crucial for us patients. But that's not necessarily the case, and I think she will tell you exactly what it is. Thank you.
Thank you very much. Can you hear me okay? Yes? Okay. Good morning, and thank you for this opportunity. It's really great to be here. I think that the presentations that we heard previously are a really great segue to what I want to touch upon today, and really give you a little bit of an overview of what what we do, what we consider when we try to really kind of guide the drug development process and also the regulatory review of products for rare diseases, and specifically for MPS disorders. This is my disclaimer. I'm supposed to show that. So this is my outline. My goal today is not to really go into details or nuances of the laws and regulations. That's not really why I'm here, but I just want to mention a few key points and considerations that that we really all need to keep in mind, and try to really convey how FDA thinks and how we think in our division, specifically in rare diseases, where really the perspective is very, very different than when we really look at drug development in more common diseases like diabetes or hypertension. So I'll touch upon some of the regulatory fundamentals, how we apply some of the regulatory standards in the drug development and review for rare diseases, some of the challenges in designing and executing good clinical trials for rare diseases. I will touch upon a little bit about the benefit-risk framework. That's really the fundamental piece when it comes to regulatory review. And then just a little bit on patient experience data that was previously mentioned. So starting with this very basic slide, it's just the traditional kind of drug development paradigm that we follow. It starts from discovery, chemical synthesis of a product. Non-clinical refers to animal studies, which are always required and very helpful in identifying safety toxicities primarily before going into humans. So IND or investigational new drug applications are required in this country for any investigational product, and those go through a review by FDA for every product that is intended to be tested in humans. So the phases of development in general are kind of separated into this phase 1, phase 2, phase 3, just really based on the goals of those studies. And this is really depicting the traditional drug development, mostly for common diseases and in general. I have to say that what we see in rare diseases and in disorders of metabolism, there's kind of this graying of those separations where sometimes we really are not able to conduct a phase 1 study in healthy volunteers due to specific considerations of the drug. Sometimes because of the constraints of the small sample size, those are rare diseases, we're not really able to separate a phase 2 study where you explore a dose from a phase 3 study, so there's kind of this gray area. But this is kind of the general approach that follows. Once this is done, then you come and you collected the data from all the clinical trials, your animal data, chemistry, manufacturing information. and sponsors, what we call sponsors or companies, come to FDA and submit what we call an NDA, so a new drug application, or a BLA or biologics license application if it's a biologic, which is basically the whole application that is reviewed by the agency to make a decision of whether a drug should be approved or not. And then after that, you get into the post-marketing setting where the drug is really approved and out there, and data collection is continued for various reasons. So really, the overarching goal of drug development, it's very simple. It really is to gather evidence that basically establishes that a drug or a biologic is safe and effective for a specific disease. And that's really the best access to treatments, is to have an approved drug on the market. So now, building upon the previous presentations, which were really outstanding, I think, and a really good segue into what I want to present today, is really how do we define this efficacy or effectiveness, which is really a treatment benefit. So the way FDA thinks of this is really what it comes down is to how patients feel, function, or survive. And we use this a lot to really describe how we think about really providing an evidence that a treatment is beneficial. So this evidence is somewhat more straightforward when you think about survival, right? So a good example is Pompe disease, where you have the infantile form, and you show in a clinical trial that patients who are actually treated with the enzyme replacement live longer. So really, nobody can question that. That's a very powerful evidence of treatment benefit. More often than that, though, what we are interested in looking at how patients feel or function, right, in their daily life. So how do you capture that? It's very challenging, and especially in rare disease, and I'll talk about it a little bit later. But these 4 classes of what we call clinical outcome assessments, which were described a little bit earlier, is different ways or instruments to really capture that experience. So it's from patient-reported, so patients are the ones who report their symptoms and changes in those. Clinician-reported, so investigators, physicians who are actually observing and reporting those. Observer-reported outcomes are really what it says. So the observer, which is oftentimes a caregiver or parent, make note of symptoms, how patients feel or function, and changes in those in a trial. And then performance outcomes are really functional outcomes. A good example of that is the 6-minute walk test, which has been used for many approvals of enzyme replacement therapies. Now, how do you demonstrate that? So what do we really look at? And that's just a kind of general, really bottom line from an FDA perspective. So, of course, establishing that a drug is effective is a prerequisite, is a requirement by the law. So, and that makes a lot of sense. You wouldn't want to expose a patient to a drug who potentially can have safety risks when it's not effective. Now, how do you establish that? And this is what we call the substantial evidence. So this is the bar that somebody needs to prove that a drug is truly efficacious for a particular disease. This is, very well described and actually in a lot of detail in the law, believe it or not. And so this is an excerpt from the Code of Federal Regulations, and I'm not going to go into detail, but basically what it describes is that this substantial evidence means that it is scientific evidence, data coming from adequate and well-controlled investigations. Traditionally, in common diseases like diabetes or others, the FDA in other divisions has required that companies conduct at least 2 of those clinical trials, and we call those phase 3 clinical trials, with particular predefined statistical analysis plans and expectations. We know that's not feasible in rare diseases, so how do we apply that? This is where this regulatory flexibility comes in, which is really what is kind of the bread and butter of drug development and regulatory review in rare disease. So again, very well described and in detail in the laws that while all of those standards apply to drugs, there is an understanding that there are a wide range of uses for those drugs, and there has to be some flexibility in applying those standards. And so there has to be a scientific judgment to determine the type of data and information that the agency would need for approval of a particular drug. So what really that means that instead of this paradigm of you need to conduct at least 2 trials, we understand that there's not enough patients to do that. And so what FDA recognizes is that this, what we call substantial evidence, can be demonstrated through one study, which has to be adequate and well controlled, and I'll go into a little bit of detail of what that means, plus some additional evidence to give a little bit more convincing evidence that really the data is reliable and useful. So this is just an example that I obtained from one of my colleagues. It just shows this flexibility in practice for a 7-year period for rare disease applications. Those flexible development approaches were applied to about 80% of programs, whereas compared to non-rare disease applications or your common diseases, it is less what we call this traditional development approach. So I spoke a little bit about these adequate and well-controlled studies, and this is something that is in the law, obviously, but it makes a lot of sense. And basically what this means is that the studies or clinical trials that one conducts for a particular drug have to generate data that enables you to understand and distinguish the effect of the drug from something else. So when it comes to that, we think about spontaneous change, so change that's due to the disease itself, or placebo effect. The placebo effect is more common in other diseases, and specifically for psychiatric diseases and others, not so much in our area, and also biased observations having to do with how really you assess those changes. The elements of those adequate and well-controlled studies, these are all very specific of how really we define that. The asterisks here and the underlines are put just to point out that which ones really pose a challenge in rare diseases, and particular MPS disorders, and I'll talk about each one in a little bit more detail. So when it comes to this one adequate and well-controlled study, so we often have one chance at getting this right, and this is very important. It requires a lot of preparation, a lot of collaboration, and this is what we routinely do with sponsors or companies, investigators who come to FDA to talk with us in meetings, is we discuss every aspect of the drug development, and it has to do with the rare disease and the challenges. So you see here those 5 main challenges that we deal with in the rare diseases, having to do with choosing an appropriate control group, because at the end of the day, you really need to do a comparison. How do you assign to drug versus control? How do you select those patients who are actually going to participate in the trial? How do you have measures, and what measures do you have to minimize the potential bias? We recognize there's always bias in all observations, all trials, but what can we do to at least minimize that to the degree possible? And then the last piece is having these well-defined, reliable methods to assess the treatment response. So when it comes to controls, this is a very important issue, and one that we talk about a lot with investigators and with companies. We hear from patients and patient groups that having a placebo in a trial is not really potentially ethical, or not appropriate, or really not acceptable in some circumstances. I think that makes a lot of sense. And there is flexibility in having a placebo control. Of course, the ideal would be to have a trial where you have subjects get the drug and some subjects get placebo, follow them for a period of time, and then that's how you're really able to compare the data. But there are circumstances where you really cannot do that. So there's other ways of choosing a control group. For example, if there is a current standard of care or other treatment, you can always compare to that. Or the last piece is this historical control, which is really the least favorite of all because it becomes a very big challenge when you have to really match patients and really truly understand if you're really comparing apples to apples or really something different. But we have certainly seen that happen. There's been drugs approved based of what we call a historical control group. And that's really a group of patients that are kind of obtained from history or from natural history data, natural history studies, or even retrospective data review. I have to say that becomes very challenging and always have to go into a lot of details of that ahead of time, even before the phase 3 trial starts. The next one is how do you select the patient population? So this is one that we've seen actually some programs fail due to just not choosing the right patient population. So, of course, you have to study in the patient population that the drug will be marketed for or approved for, either population or subpopulation patients. Is it going to be adults or children? That becomes really different in terms of regulations also and research in children, but also how long you're going to follow them for and what symptoms you're going to be looking at. Or some of this can be at different ages. One important point to make here is selecting the most informative group. So it could be that a drug, for example, is approved for a specific disease, let's say MPS I. In MPS I, though, there is a very severe form, which you see that there's a very much more rapid rate of progression. In terms of really showing a treatment effect in the constraints of a clinical trial, which is at most 1-2 years if you're lucky, that may be the most informative group because you would expect to see changes. If it is that you're potentially enrolling somebody who has very mild disease, would you expect to see any change within a year or two? Maybe not, and so they may not be the most informative group, at least for the purposes of showing a treatment effect in a trial. And then I also want to stress what Dr. Shapiro and others stress is the natural history. It becomes very critical. So in terms of what symptoms the subjects should have a baseline so that you can actually show change, are those clinically meaningful? How fast do they progress? Are there any markers that could potentially predict the rate of progression, such as their genetic mutation status? And in some diseases that's available, and in others is not. And are there subpopulations that may have greater disease severity, as I previously talked about, or faster rate of progression? Now, in terms of minimizing this bias, of course, the ideal scenario is that you randomize patients to a drug or control. There's a lot of reasons to do that, and the main reason being that you want to create almost like a balance in the baseline characteristics of that group, where when it comes to comparisons, you have a confidence that you are actually comparing similar groups. And the only difference in our groups should be the treatment, meaning the drug. Now, there are baseline characteristics that you know about. For example, patients who may have a null mutation in MPS I or others tend to be more severely affected or have a rate of progression that's more severe. That's something that you potentially know. But there's many others that are unknown. Randomization ideally takes care of that as well. So known and unknown imbalances. And that's why we always try to push for that. Blinding, because that can introduce bias when it comes to either patient investigators or sponsors or clinician or observers who actually fill out the questionnaires or actually have to make a judgment on the patient changes on treatment. There has to be, of course, a standard protocol that's been followed to make sure you don't introduce a bias. The trial duration is one that's important. So, for example, if you're looking at what we previously talked about, cognitive function, neurologic effects, if you have a 6-month trial, chances are you're just not going to see anything. And so when a sponsor, for example, comes in and their goal is to really stabilize the disease, well, from 0 to 6 months, nothing changed. Does that mean that you stabilize things, or is it just that what happened anyway? So adequate trial duration becomes so fundamental when it comes to especially neurologic changes or cognitive changes, and that can certainly introduce bias. And lastly, the data quality. Did folks really collect the data they're supposed to at the intervals they're supposed to? A big problem becomes missing data in our observations, where we don't really know how to interpret, and that really decreases our ability to really make good interpretations. And we know we don't have a lot of patients anyway, so the more data you're missing, the less data you have from a very, very restricted pool of patients. Now, the last piece is this well-defined, reliable methods that really applies to how do you capture and how do you describe and measure this treatment response. That has to do with these clinical outcome assessments. So again, the natural disease progression, natural history is really the most important thing. A lot of times we have sponsors who come in and they want to study a disease, they have a product in mind, and then we talk about, well, what are you going to really look at? Which endpoints are those meaningful to patients? Have you talked to patients? And we end up being in a place where, well, no, there's limited information because it's a rare disease. Well, that's not really acceptable, right? I mean, it is a rare disease, we understand that, but you have to do your research, you have to do your homework. If you embark on a whole clinical development program without knowing the natural history of this disease, it's more likely than not that's going to fail. So when it comes to clinical outcome assessments, I briefly touched on that, is those instruments that we use that really mention this feel, function, and survive, primarily the feel and function. So this is just a list of additional considerations. I'm not going to spend a lot of time. I think I'm kind of preaching to the choir here. Everybody's very familiar and aware of those considerations, and we're for sure very familiar, and we deal with those day to day when it comes to rare disease drug development. Now, just very briefly, and I know I'm out of time, I wanted to discuss this benefit-risk framework. So when it comes to regulatory review, when a marketing application comes in, the whole framework that the FDA and our division really operates under is understanding what are the benefits. So that basically is the data that shows you how the drug works. What are the risks? Every drug has risks, no matter how small or big. How do you manage those risks? And all of that has to go into the context of the particular condition and any other available treatments. So this is what is called this benefit-risk framework, which becomes really important in rare diseases, and that's where knowing the pathophysiology of disease and natural history is, without saying, really a very important piece. And the last piece I want to touch upon is very much highlights of this patient-focused drug development that was really started back in 2012 and reaffirmed with 2017 laws, is how do you capture this patient perspective experience? And we know for a fact that we are very interested in that, and how do you incorporate it in the regulatory review and decision making? There's been a series of public meetings that took place. One specifically had to do with neurologic manifestations of inborn errors of metabolism in 2014, where patients, parents came to White Oak campus and really talked about their experience and what outcomes are important to them, and how and what kind of treatments are they looking for. And so that was back in 2014. There was this voice of the patient that was generated that's really a summary of all of those. That actually was extremely useful when we review those applications or even provide guidance to sponsors. And the last piece is, as was previously discussed, with the 21st Century Cures Act and then the reauthorization in 2017. So basically, the regulators and the lawmakers understand that the patient voice is very important, and it needs to be incorporated to some degree, but with some rigor within a regulatory review. I wanted to show just one of the first guidances that came out just recently in June of this year, where has to do with how do you collect this comprehensive and representative input of the patient voice. This is one of 4 guidances. There's 3 more that will be coming out that are very detailed and kind of explains the thinking behind how do you collect this data and how do we think about incorporating those in the drug development. These are just some resources for people if you're interested in reading more about it. And this is just my take-home points that you can read, but basically what I wanted to stress is the natural history information is fundamental. And really the challenges for us in interpreting this data in rare diseases come from just difficulties with designing and executing those adequate and well-controlled trials. And lastly, this patient experience data are just really increasingly becoming more and more incorporated. There's a lot more to come. This is just the beginning. But just wanted to point out that as previously discussed, we at the FDA, and of course me personally, are very interested in hearing really what is meaningful to patients and what really they're looking for when new drugs come to market or even prior to that. Thank you very much.
Thank you very much, Dr. Smpokou. And before you all leave this refrigerator to get some fresh and warm air or some coffee, are there any questions?
Can I ask a question?
Sure.
Okay.
You might have to go to the mic.
I can come up here. Right. I have a question for Dr. Smpokou. So we have often very heterogeneous groups of patients in rare diseases. And you have children at different rates of progression or different levels of progression. You have different ages, you have different mutations, and somehow you have to put them all into some kind of clinical trial. Do you think that there's room for a sort of individualized patient outcome measure where you would sort of set in advance what would constitute for each patient a success based on what their level of performance is, what their parents want, what, you know, sort of a dimensional kind of outcome for a very small group of patients. Do you understand what I'm saying?
Yeah, I totally understand. So that's a good question. That's one actually that we oftentimes talk about and struggle with. And so I guess there's 2 different points to make on that. One is that it's good to include the most patients that you can, of course, in a clinical trial because you don't have that many. The natural history information, of course, is fundamental to really identify are there predictors or are there certain ages or baseline functions that kind of really subcategorize the patients. There's ways to deal with that in terms of statistical approaches where you really analyze groups separately or differently and stratifying. That's one. Another is what you're describing is kind of these individualized endpoints based on what a patient comes in with in the trial and at what stage of the disease. If you have a very good understanding of the progression of its subgroup, I think it's reasonable to really individualize for groups of patients. But this information from natural history, of course, has to be very detailed, right, and very, very important. So I think that's very reasonable, and we always welcome discussions about that. The other point to make on that is you may not necessarily, in a given disease, want to recruit every single patient. So that comes down to what I call this informative population. So when you have a clinical trial, you're dealing with rare disease, whether it's 50 versus 20, you know, it's small numbers we're dealing with, you know. And so there's usually this kind of global approach on looking at everything, this totality of the data that we always look at. So is there a chance that you can just isolate the most informative population for study in a trial in which you're able to really conclusively show treatment difference? And then your supportive evidence can come in other populations that are studied either concurrently or separately, and maybe in a different way to supplement that. So now the disease biology and the basic single enzyme defect and how the pathophysiology works, actually, we take that in a lot of consideration when we look at this data. And so does it make biologic sense, right, in some patients that you would see this effect? So that becomes important. So I would say there's different ways to deal with that. We always encourage investigators and sponsors to come talk to us very early because it can be disease specific. But I think there's creative ways to really overcome that in those heterogeneous diseases.
Thank you very much. Any other questions? Yes, ma'am?
I have a question. You talked about how a 6-month trial with a neurocognitive endpoint, for example, might not be long enough to see an effect. And we've all heard Commissioner Gottlieb talk a lot about accelerating product development for rare diseases. Are you able to talk about how the agency is thinking about ways, particularly for MPS diseases or rare diseases like these lysosomal storage diseases, where how to accelerate product development and still have endpoints that are meaningful, be it biomarkers or patient data, and really to assess benefit risk to bring these products forward more quickly?
Yeah, sure. That's a good question. And that's very difficult. What I would say that a lot of preparation goes into it. I think that we see a lot of times companies come to us and they're really eager to go into their, phase 3 trial and really prove, which of course we want to see. But what people don't realize is the preparation that has to go in even before then. And so one aspect would be really doing a good natural history study or obtaining the most information that you have even before thinking about a trial. So a 6-month trial may be reasonable depending on what assessments you're looking at, specifically about neurologic function in a particular disease. And Dr. Shapiro will tell you, that takes a long time. So even if you have all the data that you can for 6 months, how likely is it that you could really change unless you really have a very severe disease where those patients are at a point where they rapidly decline. And again, that's very disease specific. So of course, the accelerating drug development, that's everybody's goal. There's different ways to do that. There's different pathways to go through that where you get kind of approval ahead of really completing a full trial with clinical outcomes, and those details have to be really discussed within the concept, the constraints of a specific product. But what I would say is oftentimes what we see is we want to accelerate drug development. We want to have the shortest trial possible, which makes a lot of sense. But of course, you don't want to set yourself off for failure if you don't have enough information when you go into it. So, for example, could it be that, 6 or 8 months is enough to show changes in sleep, to show changes in behavior? That's conceivable, but have you looked at that before? What does this disease look like? What are the patients are telling you? And so if that's the case, we certainly wouldn't object to it. But I think the bottom line becomes your duration of the trial really needs to be informed by what happens in the disease and over what period of time. So I don't know if that makes sense.
Yeah. Just
one quick
one
comment
about that, and that is that if you can somehow secure a sample that is just precisely at the inflection points of decline, for example, in MPS 1, a group of patients between ages 1 and 2, you might be able to show in 6 months a change because you do have that really massive change in development. But I can't imagine that there are many diseases that you might be able to do that with. Maybe MPS 3, but you would have to know exactly by mutation what kind of progression is expected. Right.
Dr. Smpokou, I just want to mention one quick thing. The FDA came out with a request for information a couple of days ago on COAs. So I want to make sure this audience is aware that there's now a request out, right, to get information from the public on the clinical outcomes assessments, which is, I think, a great opportunity for this audience to contribute input.
Yeah, that would be awesome. Yeah. Thank you.
Hi. All of the clinical trials that I have heard about are very specific to age groups, as opposed to how the patients, where they are in the progression of the disease. Having 2 adult children with MPS IIIB, they haven't qualified for any of the clinical trials because they are so much older, and yet their abilities are more than some of the very, very young patients. So I wondered if clinical trials ever are done based on where the patients are in the progression of the disease, as opposed to just an age group.
Is that a question for me?
I don't know.
So I can. So that's also a good point. I would say that, that what you're talking about is this kind of eligibility criteria for trials that I would say largely, companies define that, and sometimes they talk to FDA about, sometimes not. We oftentimes provide recommendations for those. The other point to make is that I guess it depends on what you're actually looking at. So in adults, there are different challenges with the disease, as we've heard about, and in kids, there are different challenges. And so does that mean that the challenges that an adult faces are not meaningful? Of course not. And so if a trial includes some of those patients and look at those separately, but look at separate types of endpoints that really make a difference in somebody's life, I don't know that that would be a problem. I think, the general approach is you want to look at something that really changes somebody's daily function, right, or how they really feel and function. So if there's ways to capture that reliably, and if there's ways to incorporate that in the trial and follow that over time, that sounds like it would be clinically meaningful. Now, in terms of the eligibility criteria, I would say that's something that companies a lot of times make a decision on, maybe depending on the availability of patients or other maybe feasibility concerns. But from, I guess, an agency perspective, we can provide guidance in that and kind of stimulate conversation in that. But I think your point is very well taken, and it makes a lot of sense.
Can I just say one other thing about that, and that is that in older patients, one of the problems is that if you're looking at disease progression, it's much slower than it is with younger children. And so the companies don't want to have a 5-year trial period to see what happens with a 20-year-old, let's say, or even a 15-year-old. They would rather take a younger group where you know what the progression is like based on mutation or other kinds of factors, and see a more rapid response. But that isn't to say that one can't do that, but I think that the outcomes need to be different, as you said.
Yeah.
With the natural history information that's available, we don't have the breadth of natural history information or long-term natural history information on slower progressing forms of the disease. So that's a real need that's out there.
That's
Just
correct.
the only thing I wanted to add actually is that we're talking strictly about clinical trials investigations, right? And so there's a separate type of INDs that we call expanded access, right? So those are really truly for treatment. So those are for serious life-threatening diseases. Of course, rare diseases, MPSs would be serious and life-threatening, and there's some other criteria. But sometimes when we deal with very rare diseases, one good example is MPS 7, where a lot of expanded access data was actually considered in the process of the review because there's really not enough patients. If there's good data collection as part of those treatment type of INDs in adults, I think that's also a good way to go.
Yeah, the parent MPS II post BMT transplant. That being said, how do you base the longevity of a clinical trial? How do you determine the termination of the timeline?
Well, I can talk to that. You're talking about MPS II, is that?
I'm talking about any
yeah.
kind of
Well, I mean, I
clinical
think it
trial.
depends on the rate of progression of the disease and your sample. And so, yeah, one of the real challenges, and MPS II is an example, is that there's so much variability in the rate of progression that it's really hard to determine what a length of a trial ought to be. It's much easier in MPS III or MPS I, where you have a better idea about what that rate is going to look like and whether you're going to be able to see change. MPS II is really a challenge because of that, and because we don't have good predictors of, you know, the neuronopathic and the non-neuronopathic forms. Yeah, I think we probably have to abandon
one
ship.
last question, please.
Hi, sorry, it's not about a study. I'm a mom of 2 kids with San Filippo, and I just have questions, and I need help. I need help on how to educate schools and places. I have a son with serious behavioral problems, and my son gets burned. He comes home with bruises, and I'm always told, oh, he just ran into the wall or he tripped, and you don't know because they don't speak. So I need to educate people about the behaviors, or I'm told he's pushing, he's doing these things. Sometimes they are, they're not, you don't know. I don't know if there's something out there to teach people about these behaviors, that they're not bad kids. They're sick. They have a disease, and people don't get it. So I wanted to know any ideas of literature that I could give people to educate. And secondly, outside of drugs, which ain't working, if there's any other ideas to help with these behaviors, because they do make everyone's quality of life difficult. So I was hoping somebody had ideas, because at home, I mean, I have a great doctor who's never heard anything about this, and so he told me, you bring me ideas, and I will write you what you want. I will send you wherever. But he's a nice guy. I'm not sure he's really wanting to learn much about this. You know, I don't know of anyone, at least locally, with the disease my children have. And so I'm trying to get ideas so I can give professionals what to do. Help me so I can help my family. I have 2 kids. One was very low functioning, one's very high functioning. And the high functioning one is kind of just floating along in the outskirts, in the shadows of the one who gets all the attention. So any help from anyone up there would be great.
Oh, I feel like I could, I don't want to step in before you. I do want to mention this really incredible resource. It's a national organization that I just became very familiar with as part of a working group on newborn screening for the Minnesota Department of Health. They're called Parent to Parent Voice. They are, you know, we should talk afterwards so I can give you the website because I don't have it memorized. But it is an amazing program. It's a nonprofit, and they have really structured education on empowering parents and supporting parents in advocacy, advocating for their child, and it is specifically for parents who have children with multiple medical and developmental needs. And they are fantastic. There are parents of children with other lysosomal diseases who attested to the degree of support that they got. And there is a really rigorous training program involved in partnering you with an advocate. And they have it tailored to what your needs are. So that's one really structured form of support. I'd be happy to talk with you after about some of the recommendations that we make at our center. And I'm sitting next to a complete expert here, so.
Thank you for sharing your struggles, and a lot of us in the room can absolutely 100% identify with everything that you're saying. So know that you are not alone in feeling that way and experiencing those things. I'm happy to talk with you afterwards. I'm a pediatrician by training, but do Sanfilippo full-time now, and I'm also a parent to a little girl with Sanfilippo. So I understand where you're coming from. There is a great need for those educational resources and helping people to understand the unique challenges that our kids have and how to interpret our behaviors in a way that don't discriminate against our children, and provide them the best care possible. So while I don't have specific answers to your individual situation, I'm happy to help you, outside of here as well.
Okay. Thank you very much. We shall reconvene in about 10 minutes' time, just before 10 o'clock, to continue the next session. Thank you.